Bacteriology. How to characterize bacteria. Staining, a process of artificially coloring microorganisms with dyes in order to facilitate their study under the microscope. Dye, also known as stain. Smear, a thin layer of material spread across a slide for microscopic study. It usually contains the microorganisms to be studied or observed. Fixation, the process of fixing microorganisms or killing the microorganisms and attaching them to the slide. Mordant, added to the primary dye to intensify the primary stain. It increases the affinity of the stain to the biological specimen. It tends to coat structures and makes it thicker and easier to stain. Grimm's stain is one of the most frequently used processes in identifying bacteria and is used daily in hospitals. It is a primary test which quickly and cost-effectively divides bacteria into one of two types, gram-positive or gram-negative. Gram-positive bacteria have thicker cell walls with less fatty substances, lipids, than gram-negative bacteria. When washed with solvent, the cell pores close, becoming less permeable and are able to retain the stain, in this case, purple. Gram-negative bacteria have thinner cell walls with more lipids. The solvent dissolves the lipids, which combined with thinner cell walls, washes out or decolorizes the stain. In order to see the decolorized bacteria, a counter stain, safranin O, is added to exaggerate the contrast with gram-negative cells. Acid-fast stain. It is the differential staining techniques which was first developed by Zeil and later on modified by Nielsen. So this method is also called Zeil-Nielsen staining techniques. Nielsen in 1883 used Zeil's carbylfuchsin and heat, then decolorized with an acid alcohol, and counter-stained with methylene blue. Thus Zeil-Nielsen staining techniques was developed. The main aim of this staining is to differentiate bacteria into acid-fast group and non-acid-fast groups. This method is used for those microorganisms which are not staining by simple or gram staining method, particularly the member of genus Mycobacterium, are resistant, and can only be visualized by acid-fast staining. The endospore stain is a differential stain used to visualize bacterial endospores. Endospores are formed by a few genera of bacteria, such as Bacillus. By forming spores, bacteria can survive in hostile conditions. Spores are resistant to heat, desiccation, chemicals, and radiation. Bacteria can form endospores in approximately 6 to 8 hours after being exposed to adverse conditions. The normally growing cell that forms the endospore is called a vegetative cell. Spores are metabolically inactive and dehydrated. They can remain viable for thousands of years. When spores are exposed to favorable conditions, they can germinate into a vegetative cell within 90 minutes. Capsule stain is a type of differential stain which uses acidic and basic dyes to stain background and bacterial cells respectively so that presence of capsule is easily visualized. Capsule is synthesized in the cytoplasm and secreted to the outside of the cell where it surrounds the bacterium. Negative staining is an established method, often used in diagnostic microscopy, for contrasting a thin specimen with an optically opaque fluid. In this technique, the background is stained, leaving the actual specimen untouched, and thus visible. Spirochetes are spiral or corkscrew-shaped bacteria. These organisms are the cause of syphilis and Lyme disease. Silver stains are typically used to identify spirochetes, but Giemsa can also be used as a quick screen for microorganisms. Warth and starry silver stain demonstrates spirochetes by staining them black. Major groups of clinically significant bacteria. 1. Thick-walled. Gram-positive cocci and bacilli. Gram-negative cocci and bacilli. Rickettsia and chlamydia. 2. Thin-walled. Spirochetes, Treponema, Leptospira, Borrelia. 3. Wall, less. Mycoplasma. Ureaplasma. Gram-positive cocci. Staphylococci. Staphylococci are gram-positive aerobic organisms. Staphylococcus aureus is the most pathogenic. It typically causes skin infections and sometimes pneumonia, endocarditis, and osteomyelitis. It commonly leads to abscess formation. Staphylococcus aureus is a non-motile, gram-positive, catalase-positive, and facultative anaerobic coccus. The bacterium occurs widely, in low numbers, on the skin and mucous membranes of warm-blooded animals. Normally, the bacterium is transient and not a part of the resident flora on skin. Staphylococcus epidermidis is a very hardy microorganism, consisting of non-motile, gram-positive cocci, arranged in grape-like clusters. It forms white, raised, cohesive colonies about 1 to 2 mm in diameter after overnight incubation, and is not hemolytic on blood agar. Staphylococcus saprophyticus is a coagulase-negative species of Staphylococcus. Like other Staphylococci, it is gram-positive, is globular-shaped, and is a facultative anaerobe. It has abundant transporter systems to adapt to ever-changing pH, osmolarity, and concentration of urea in human urine. Streptococcus. Streptococci are gram-positive, non-motile, non-spore-forming, catalase-negative cocci that occur in pairs or chains. Older cultures may lose their gram-positive character. Most streptococci are facultative anaerobes, and some are obligate, strict, anaerobes. Most require enriched media, blood agar. Species of Streptococcus are classified based on their hemolytic properties. Alpha hemolytic species cause oxidization of iron in hemoglobin molecules within red blood cells, giving it a greenish color on blood agar. Beta hemolytic species cause complete rupture of red blood cells. On blood agar, this appears as wide areas clear of blood cells surrounding bacterial colonies. Gamma hemolytic species cause no hemolysis. Beta hemolysis, sometimes called complete hemolysis, is a complete lysis of red cells in the media around and under the colonies. The area appears lightened, yellow, and transparent. Streptolysin, an exotoxin, is the enzyme produced by the bacteria which causes the complete lysis of red blood cells. There are two types of streptolysin, streptolysin O, slow, and streptolysin S, SLS. Streptolysin O is an oxygen-sensitive cytotoxin, secreted by most group A Streptococcus, gas, and interacts with cholesterol in the membrane of eukaryotic cells, mainly red and white blood cells, macrophages, and platelets, and usually results in beta hemolysis under the surface of blood agar. Streptolysin S is an oxygen-stable cytotoxin also produced by most gas strains which results in clearing on the surface of blood agar. SLS affects immune cells, including polymorphonuclear leukocytes and lymphocytes, and is thought to prevent the host immune system from clearing infection. Streptococcus pyogenes, or gas, displays beta hemolysis. Gamma hemolytic species cause no hemolysis. Streptococcus pyogenes is a species of gram-positive, aerotolerant bacterium in the genus Streptococcus. These bacteria are extracellular, and made up of non-motile and non-sporing cocci. It is clinically important for humans. It is an infrequent, but usually pathogenic, part of the skin microbiota. Streptococcus agalactiae is a gram-positive coccus that, when cultured on sheep blood agar, forms glistening gray-white colonies with a narrow zone of beta hemolysis. It is an invasive encapsulated organism capable of producing severe disease in immunocompromised hosts. Streptococcus bovis is a gram-positive bacterium that grows in pairs or chains of cocci and is a normal inhabitant of the gastrointestinal tract. It is both oxidase and catalase negative and is a non-motile, non-sporulating, facultative anaerobe. The viridans streptococci are a large group of commensal streptococcal gram-positive bacteria species that are alpha hemolytic, producing a green coloration on blood agar plates, hence the name viridans, from Latin virides, green. Streptococcus pneumoniae cells are gram-positive, lancet-shaped cocci, elongated cocci with a slightly pointed outer curvature. Usually, they are seen as pairs of cocci, diplococci, but they may also occur singly and in short chains. When cultured on blood agar, they are alpha hemolytic. Gram-positive bacilli, rods, are divided into their ability to produce spores. Bacillus and Clostridia are spore-forming rods while Listeria and Corynebacterium are not. Spore-forming rods that produce spores can survive in environments for many years. Anthrax is caused by Bacillus anthracis. Humans acquire the disease directly from contact with infected herbivores or indirectly via their products. The clinical forms include 1. Cutaneous anthrax, eschar with edema, from handling infected material, this accounts for more than 95% of cases, 2. Intestinal anthrax, from eating infected meat, and 3. Pulmonary anthrax, from inhaling spore-laden dust. There are 3 forms of naturally occurring human anthrax infection. Cutaneous anthrax is the result of spores entering the body through small breaks in the skin. This form of the disease is characterized by a sore at the point of infection that develops into a painless ulcer covered by a black scab, eschar. Cutaneous anthrax accounts for approximately 95% of all reported human anthrax cases. Cutaneous anthrax could also occur as a result of an aerosol attack. Inhalational anthrax is the result of breathing B. anthracis spores into the lungs. Inhalational infection is the form of anthrax that would be of most concern following an intentional aerosol attack with B. anthracis. Gastrointestinal anthrax typically occurs as a result of eating the meat of animals infected with B. anthracis. The intestinal tract, mouth, or throat, or a pharyngeal anthrax, may be infected. 1 GI anthrax is normally thought to occur as a result of ingestion of vegetative bacteria rather than spores, therefore, GI anthrax is not expected to result from exposure to aerosolized spores. Bacillus cereus. The bacteria is classically contracted from fried rice dishes that have been sitting at room temperature for hours. Bacillus cereus bacteria are facultative anaerobes, and like other members of the genus Bacillus, can produce protective endospores. Its virulence factors include cerealicin and phospholipase C. Bacillus subtilis is non-pathogenic but can contaminate food and be considered an opportunistic pathogen among the immunocompromised. They are used on seeds, vegetables, and plants as a fungicide because of their ability to produce antibiotics. Bacillus subtilis inhabits the root system of the plant, completing with disease-causing organisms. Some Bacillus subtilis strains are capable of producing toxins for insects. These strains are used by farms to protect their crops. Clostridium, genus of rod-shaped, usually gram-positive bacteria, members of which are found in soil, water, and the intestinal tracts of humans and other animals. Most species grow only in the complete absence of oxygen. Dormant cells are highly resistant to heat, desiccation, and toxic chemicals and detergents. Botulism is a disease caused by this neurotoxin, specifically A, B, E, or F-type neurotoxin. Symptoms include a flaccid paralysis, weakness or slackness, in various muscles. Bacteria called Clostridium botulinum produce the neurotoxin. The neurotoxin paralyzes muscles and can be life-threatening. Tetanus is an acute, often fatal, disease caused by an exotoxin produced by the bacterium Clostridium tetani. It is characterized by generalized rigidity and convulsive spasms of skeletal muscles. The muscle stiffness usually involves the jaw, lockjaw, and neck and then becomes generalized. Clostridium perfringens food poisoning is acute gastroenteritis caused by ingestion of contaminated food. Symptoms are watery diarrhea and abdominal cramps. Gas gangrene and clostridial myonecrosis are interchangeable terms used to describe an infection of muscle tissue by toxin-producing Clostridia. Clostridioides difficile, also known as C. diff, is a bacterium that causes diarrhea and colitis, an inflammation of the colon. Gram-positive non-spore-forming rods. Corynebacterium diphtheriae is the leading causing agent of diphtheria. It is a non-motile, non-encapsulated, non-sporulating gram-positive rod-shaped bacterium with a high GC content and occurs in 4 biovars, Gravis, Mitis, Intermedius, and Belfinti, based on colonial morphology and biochemical profiles. Propionibacterium acnes is a gram-positive human skin commensal that prefers anaerobic growth conditions and is involved in the pathogenesis of acne. Listeria monocytogenes is a gram-positive bacterium, in the division Firmicutes, named after Joseph Lister. Its ability to grow at temperatures as low as 0 degrees Celsius permits multiplication at typical refrigeration temperatures, greatly increasing its ability to evade control in human foodstuffs. Erysipelathrix regiopathiae is a gram-positive, catalase-negative, rod-shaped, non-spore-forming, non-acid-fast, non-motile bacterium. Distributed worldwide, Erysiopathiae is primarily considered an animal pathogen, causing the disease known as erysipelas that may affect a wide range of animals. Actinomyces israelii is a species of gram-positive, rod-shaped bacteria within the Actinomyces. Known to live commensally on and within humans, A. israelii is an opportunistic pathogen and a cause of actinomycosis. Nocardia asteroids species are filamentous aerobic, gram-positive, and mycolic acid cell wall bacteria. Nocardia colonies vary from white to tan, orange and red in color. They have a lipid bilayer cell membrane with associated proteins. Lactobacillus acidophilus is a type of bacteria found in your intestines. It is a member of the Lactobacillus genus of bacteria, and it plays an important role in human health. Lactobacillus casei is a species of genus Lactobacillus found in the human urinary tract and mouth. This particular species of lactobacillus is documented to have a wide pH and temperature range, and complements the growth of Lactobacillus acidophilus, a producer of the enzyme amylase, a carbohydrate-digesting enzyme. The most common site of Neisseria gonorrhoeae infection is the urogenital tract. Men with this infection may experience dysuria with penile discharge, and women may have mild vaginal mucopurulent discharge, severe pelvic pain, or no symptoms. Other Neisseria gonorrhoeae infections include anorectal, conjunctival, pharyngeal, and ovarian/uterine. Infections that occur in the neonatal period may cause ophthalmia neonatorum. If left untreated, Neisseria gonorrhoeae infections can disseminate to other areas of the body, which commonly causes synovium and skin infections. Disseminated gonococcal infection presents as a few skin lesions that are limited to the extremities. These lesions start as papules and progress into bullae, petechiae, and necrotic lesions. Bacteria called Neisseria meningitidis cause meningococcal disease. Neisseria meningitidis, often referred to as meningococcus, is a gram-negative bacterium that can cause meningitis and other forms of meningococcal disease such as meningococcemia, a life-threatening sepsis. Haemophilus influenza, formerly called Pfeiffer's bacillus or Bacillus influenza, is a gram-negative, coccobacillary, facultatively anaerobic pathogenic bacterium of the family Pasteurellacea. H. influenza was first described in 1892 by Richard Pfeiffer during an influenza pandemic. Haemophilus duckeri is a fastidious gram-negative coccobacillus bacteria. It causes the sexually transmitted disease chancroid, a major cause of genital ulceration in developing countries characterized by painful sores on the genitalia. Haemophilus influenza biogroup Aegyptius, HA, is a causative agent of acute and often purulent conjunctivitis, more commonly known as pink eye. Bordetella pertussis is a gram-negative, aerobic, pathogenic, encapsulated coccobacillus of the genus Bordetella, and the causative agent of pertussis or whooping cough. Legionella pneumophila is a thin, aerobic, pleomorphic, flagellated, non-spore-forming, gram-negative bacterium of the genus Legionella. L. pneumophila is the primary human pathogenic bacterium in this group and is the causative agent of Legionnaire's disease, also known as Legionellosis. Brucella is a genus of gram-negative bacteria, named after David Bruce, 1855–1931. They are small, 0.5 to 0.7 by 0.6 to 1.5 m, non-encapsulated, non-motile, facultatively intracellular coccobacilli. Brucellosis is a bacterial infection that spreads from animals to people. Most commonly, people are infected by eating raw or unpasteurized dairy products. Sometimes, the bacteria that cause brucellosis can spread through the air or through direct contact with infected animals. Signs and symptoms of brucellosis may include fever, joint pain, and fatigue. The infection can usually be treated with antibiotics. However, treatment takes several weeks to months, and the infection can recur. Brucellosis affects hundreds of thousands of people and animals worldwide. Avoiding raw dairy products and taking precautions when working with animals or in a laboratory can help prevent brucellosis. Yersinia pestis, formerly Pasteurella pestis, is a gram-negative, non-motile, rod-shaped, coccobacillus bacterium, with no spores. It is a facultative anaerobic organism that can infect humans via the oriental rat flea, Xenocilicia p. Francisella tularensis is a pathogenic species of gram-negative coccobacillus, an aerobic bacterium. It is non-spore-forming, non-motile, and the causative agent of tularemia, the pneumonic form of which is often lethal without treatment. Pasteurella multocida is a small, gram-negative, non-motile, non-spore-forming coccobacillus with bipolar staining features. Pasteurella multocida is a common cause of infection following bites or scratches caused by dogs and, especially, cats. It is rarely reported, however, and apparently often overlooked as a pathogen. The typical clinical manifestation is a rapidly developing cellulitis at the site of injury. Bartonella hensleyi, formerly Rhocolimia, is a proteobacterium that is the causative agent of cat scratch disease, Bartonellosis. Bartonella hensleyi is a member of the genus Bartonella, one of the most common types of bacteria in the world. It is a facultative intracellular microbe that targets red blood cells. Enterobacteriaceae. Gram, rods, modal W/Poritrechus flagella except Klebsiella, Shigella. Facultative anaerobe, glucose fermenters, often produce gas. Catalase, plus, except Shigella dysenteriae. Oxidase, except Vibrio. Reduce nitrate to nitrite except Erwina, Pantoa agglomerans. Escherichia coli is a type of bacteria that normally lives in your intestines. It's also found in the gut of some animals. Most types of E. coli are harmless and even help keep your digestive tract healthy. But some strains can cause diarrhea if you eat contaminated food or drink fouled water. Klebsiella pneumoniae are bacteria that normally live in your intestines and feces. These bacteria are harmless when they're in your intestines. Serratia marcescens is a gram-negative bacillus that occurs naturally in soil and water and produces a red pigment at room temperature. It is associated with urinary and respiratory infections, endocarditis, osteomyelitis, septicemia, wound infections, eye infections, and meningitis. Shigellosis is an infectious disease caused by a group of bacteria called Shigella. Most who are infected with Shigella develop diarrhea, fever, and stomach cramps starting a day or two after they are exposed to the bacteria. Salmonella infection, salmonellosis, is a common bacterial disease that affects the intestinal tract. Salmonella bacteria typically live in animal and human intestines and are shed through feces. Humans become infected most frequently through contaminated water or food. Proteus is a genus of gram-negative proteobacteria. Proteus bacilli are widely distributed in nature as saprophytes, being found in decomposing animal matter, sewage, manure soil, the mammalian intestine, and human and animal feces. Enterobacter are gram-negative bacteria that are classified as facultative anaerobes, which means that they are able to thrive in both aerobic and anaerobic environments. Many species possess flagella and thus are motile. Vibrios are gram-negative gamma-proteobacteria that are ubiquitous in marine, estuarine, and freshwater environments and encompass a diverse group of bacteria including many facultative symbiotic and pathogenic strains. Vibrio cholerae is a gram-negative, comma-shaped bacterium. The bacterium's natural habitat is brackish or salt water where they attach themselves easily to the chitin-containing shells of crabs, shrimps, and other shellfish. Vibrio parahemolyticus is a curved, rod-shaped, gram-negative bacterium found in brackish, salt water, which, when ingested, causes gastrointestinal illness in humans. Vibrio parahemolyticus is oxidase-positive, facultatively aerobic, and does not form spores. Vibrio vulnificus is a species of gram-negative, motile, curved rod-shaped, bacillus, pathogenic bacteria of the genus Vibrio. Present in marine environments such as estuaries, brackish ponds, or coastal areas, Vibrio vulnificus is related to Vibrio cholerae, the causative agent of cholera. Campylobacter jejuni is one of the most common causes of food poisoning in Europe and in the United States. The vast majority of cases occur as isolated events, not as part of recognized outbreaks. Helicobacter pylori is a type of bacteria. These germs can enter your body and live in your digestive tract. After many years, they can cause sores, called ulcers, in the lining of your stomach or the upper part of your small intestine. For some people, an infection can lead to stomach cancer. Pseudomonas aeruginosa is a gram-negative opportunistic pathogen that causes severe acute and chronic infections at different sites within the body such as urinary tract, skin, burn or surgical wounds, and the respiratory tract. The versatile, blue-green pus bacteria that opportunistically infects people, especially those who are immunocompromised. Pseudomonas rarely causes infection in healthy individuals, but it is a major cause of hospital-acquired, nosocomial, infections. It tends to infect people with immunodeficiency or burns and those with indwelling catheters or on respirators. Infection with Pseudomonas can lead to urinary tract infections, sepsis, bloodstream infection, pneumonia, pharyngitis, and many other medical problems. Pseudomonas colonizes the lungs of patients with cystic fibrosis, CF, and contributes to the chronic progressive pulmonary disease and death rate in CF. Bacteroides fragilis is an obligately anaerobic, gram-negative, rod-shaped bacterium. It is part of the normal microbiota of the human colon and is generally commensal, but can cause infection if displaced into the bloodstream or surrounding tissue following surgery, disease, or trauma. Fusobacterium is a genus of anaerobic, gram-negative, non-spore-forming bacteria, similar to Bacteroides. Individual cells are slender rod-shaped bacilli with pointed ends. Strains of Fusobacterium cause several human diseases, including periodontal diseases, Lemierre's syndrome, and topical skin ulcers. Mycobacterium avium complex, MAC, also called Mycobacterium avium intracellulare complex, is a microbial complex of 3 Mycobacterium species. It causes Mycobacterium avium intracellulare infection. Some sources also include Mycobacterium avium subspecies paratuberculosis, MAP. Mycobacterium tuberculosis, MTB, is a species of pathogenic bacteria in the family Mycobacteriaceae and the causative agent of tuberculosis. First discovered in 1882 by Robert Koch, M. tuberculosis has an unusual, waxy coating on its cell surface primarily due to the presence of mycolic acid. Mycobacterium leprae is a bacterium that causes leprosy, also known as Hansen's disease, which is a chronic infectious disease that damages the peripheral nerves and targets the skin, eyes, nose, and muscles. The Rickettsia are a diverse collection of obligately intracellular gram-negative bacteria found in ticks, lice, fleas, mites, chiggers, and mammals. They include the genera Rickettsia, Ehrlichia, Orientia, and Coxella. These zoonotic pathogens cause infections that disseminate in the blood to many organs. Coxella burnetii is a small obligate intracellular gram-negative bacterium that causes a zoonosis referred to as Q fever. The disease is prevalent worldwide and is primarily harbored by cattle, sheep, and goats. Less frequently, C. burnetii can be transmitted from cats and dogs as well as ticks. Chlamydia trachomatis, commonly known as chlamydia, is a bacterium that causes chlamydia, which can manifest in various ways, including trachoma, lymphogranuloma venereum, non-gonococcal urethritis, cervicitis, salpingitis, pelvic inflammatory disease. Chlamydia psittaceae is a type of bacteria that often infects birds. Less commonly, these bacteria can infect people and cause a disease called psittacosis. Chlamydia psittaceae is a lethal intracellular bacterial species that may cause endemic avian chlamydiosis, epizootic outbreaks in mammals, and respiratory psittacosis in humans. Potential hosts include feral birds and domesticated poultry, as well as cattle, pigs, sheep, and horses. Chlamydia pneumoniae is a type of bacteria that can cause respiratory tract infections, such as pneumonia. C. pneumoniae is one cause of community-acquired pneumonia or lung infections developed outside of a hospital. Treponema pallidum is the microaerophilic spirochete responsible for syphilis, a chronic systemic venereal disease with multiple clinical presentations. Syphilis is a sexually transmitted disease caused by the bacterium Treponema pallidum. There are 3 stages. Primary syphilis is characterized by localized disease with a painless chancre in the genital region. Secondary syphilis is characterized by disseminated disease, constitutional symptoms, rash on hands and soleus, condylomata, lymphadenopathy, and patchy hair loss. Tertiary syphilis consists of gummas, neurosyphilis with tabs dorsalis and Argyll-Robertson pupils. Congenital syphilis is a subtype in neonates acquired from the mother in the 2nd and 3rd trimesters of pregnancy, that presents with facial anomalies like snuffles, a saddle nose, Hutchinson teeth, mulberry molars, short maxillae, saber shins, and deafness due to a cranial nerve 8 palsy. Diagnosis is with VDRL or RPR, and is confirmed with FTAABS. Treatment is with penicillin G. Leptospirosis is a relatively rare bacterial infection that affects people and animals. It can pass from animals to humans when an unhealed break in the skin comes in contact with water or soil where animal urine is present. Several species of the Leptospira genus of bacteria cause leptospirosis. Borrelia is a genus of bacteria of the spirochete phylum. It causes Lyme disease, also called Lyme borreliosis, a zoonotic, vector-borne disease transmitted primarily by ticks and by lice, depending on the species of bacteria. Borrelia burgdorferi is a bacterial species of the spirochete class of the genus Borrelia. B. burgdorferi exists in North America and Europe and until 2016 was the only known cause of Lyme disease in North America. Borrelia mayonii, found in the Midwestern U.S., is also known to cause the disease. Borrelia recurrentis is a species of Borrelia, a spirochete bacterium associated with relapsing fever. B. recurrentis is usually transmitted from person to person by the human body louse. Since the 1800s, the body louse has been known as its only known vector. Mycoplasma is a genus of bacteria that lack a cell wall around their cell membranes. This characteristic makes them naturally resistant to antibiotics that target cell wall synthesis, like the beta-lactam antibiotics. They can be parasitic or saprotrophic. Mycoplasma pneumoniae is a type of atypical bacteria that commonly causes mild infections of the respiratory system. In fact, Pneumonia caused by Mycoplasma pneumoniae is sometimes referred to as walking pneumonia, since symptoms tend to be milder than pneumonia caused by other germs. The most common type of illness caused by these bacteria, especially in children, is tracheobronchitis, commonly called a chest cold. Symptoms often include being tired and having a sore throat, fever, and cough. Sometimes Mycoplasma pneumoniae can cause pneumonia, a more serious lung infection, which may require care in a hospital. Ureaplasma urelyticum infection is a type of urinary tract infection that can be sexually transmitted. It can also be passed from mother to infant during birth. It is caused by the bacterium Ureaplasma urelyticum, which can be found in a majority of sexually active people, most of whom are asymptomatic. Ureaplasma urelyticum infection is a type of urinary tract infection that can be sexually transmitted. It can also be passed from mother to infant during birth. It is caused by the bacterium Ureaplasma urelyticum, which can be found in a majority of sexually active people, most of whom are asymptomatic.